Welcome back to the NPTEL sessions of overview and integration of cellular metabolism. From the previous class we have started lipoprotein metabolism. We are going to continue the same today. So, in the previous class we have discussed the general structure and composition of lipoproteins, as well as we also have discussed the metabolism of chylomicron and VLDL, the two lipoproteins chylomicron and VLDL. Today we are going to discuss another two lipoproteins metabolism, that is LDL, low density lipoprotein, and HDL, high density lipoprotein. So, if you remember from the previous class, you have to remember because that is the continuation lipoprotein metabolism. All the lipoprotein metabolism are basically very much interrelated. So, before going through this session you need a quick recap of the previous one, mind it. So, what we have discussed regarding the VLDL metabolism, that VLDL, very low density lipoprotein, is originated from liver. VLDL is basically carrying the endogenously synthesized triacylglycerol, endogenously synthesized inside the liver actually, and it is going through different maturation phase, that is nascent VLDL as well as which has been matured to form matured VLDL. Matured VLDL after going through the capillaries is actually hydrolyzed with the help of the enzyme lipoprotein lipase. So, there is hydrolysis of triacylglycerol with this enzyme. So, finally, which is formed, that is a VLDL remnant. The remnant contains less triacylglycerol in comparison to the mature one, because the triglyceride is already synthesized. Now, we also have discussed the fate of this remnant VLDL, that is IDL or intermediate density lipoprotein. Intermediate density lipoprotein can be can enter liver, it can there is reuptake of IDL inside the liver, as well as it can form another lipoprotein, that is low density lipoprotein. So, we are going to discuss the metabolism, synthesis and fate of low density lipoprotein. So, what is low density lipoprotein? You can see this low density lipoprotein is basically the it is formed from the VLDL remnant or IDL. Now, in this low density lipoprotein, what is there? That is endogenously synthesized cholesterol in huge amount, as well as phospholipid and a very minimal quantity of triacylglycerol. And as you can see, there is one very important apoprotein, that is ApoB100. So, this ApoB100 is very specific for low density lipoprotein. Now, low density lipoprotein, it basically what it does, it transport those endogenously synthesized cholesterol from liver to peripheral or extrahepatic tissues. Now, what happens, actually this IDL is IDL means it is the VLDL remnant. VLDL remnant, again it is constantly targeted or attacked by lipoprotein lipase. So, basically there is a huge depletion of triacylglycerol. Finally, what is formed is the low density lipoprotein, which is cholesterol rich. Now, what are the fates of this LDL? Now, LDL majority, like 70% of the LDL is basically metabolized inside the liver. It enters liver through LDL receptor. The remaining 30% is basically going to extrahepatic tissue. It is, remember, it delivers cholesterol to extrahepatic tissue. So, this part is very important. LDL provides or supplies cholesterol to the extrahepatic tissue, and this uptake of LDL, say it in liver, say it in extrahepatic tissue, it occurs through LDL receptor. Now, LDL receptor is one very important receptor with respect to cholesterol metabolism. So, we are going to discuss a bit of LDL receptor as well. So, LDL receptor is also known as ApoB100 ApoE receptor. So, why it is ApoB100 ApoE receptor? Because basically LDL receptor senses ApoB100 or ApoE. So, this is the signaling molecule for LDL receptor. It is the basically acting as ligand. Ligand for this receptor is ApoB100 or ApoE. Now, this LDL receptor is present in cell surface of different types of cells like liver mainly, then peripheral tissues like adrenal glands, steroid hormone synthesizing tissues like ovary, testes, fibroblast, smooth muscle cells of vascular endothelium, lymphocytes. So, in these cells over the cell surface, LDL receptor is present in clathrin coated pits. Now, this LDL receptor, as you can see in this diagram, that it is a large transmembrane glycoprotein receptor around 115 kilodalton molecular weight. Now, this receptor has 5 domains, and the mature receptor is basically 839 amino acid long receptor containing 5 domains. So, these are our 5 domains. Now, the first domain is the ligand binding domain. The ligand binding domain is the N-terminal domain. It is situated outside the, it is actually protruded outside the cell surface. Definitely ligand binding domain, the name indicates that it binds with the ligand, that is B100 or ApoE containing lipoproteins. Now, in this ligand binding domain, there is a repeat sequence which is known as class A repeat, which contains 40 amino acids. So, there are the repeats are class A repeats containing 40 amino acids. This type of repeats, similar such 7 repeats are actually present in this domain. Now, in this domain there is cysteine, there are cysteine residues as well as acidic residues. And what is that action of this acidic residues? Acidic means amino acids which are having acidic residues. Now, those acidic residues, they interact with the basic residues of apoproteins. So, basically there is an interaction between the acidic residues of the LDL receptor with the basic residue of the lipoprotein. So, this is the reason by which ligand binding occurs. Remember, ligand is the lipoproteins, those lipoproteins which contains once again ApoB100 and ApoE. Okay. So, after that the second domain is EGF precursor homology domain. EGF is epidermal growth factor. Now, by the name it is evident that this domain, this part of the domain has sequence homology or structural homology with the epidermal growth factor. Now, this domain is important for expulsion of the attached LDL inside the cell. So, once the ligand is bound to the receptor, it also needs to deliver the ligand or lipoprotein here to inside the cell. So, this region is basically important for repulsion. So, it extrudes the bound lipoprotein to inside the cell. Next is O-linked sugar rich domain, ortho-linked sugar rich domain. There are multiple O-linked glycosidic bonds present in this domain. Glycosides are attached in this domain. So, this domain is forming a stud, is a segment which is actually holding the receptor in such a pattern that it can be expanded outside and can deliver its proper function. So, it is basically holding the receptor to the outer surface of the cell. Next domain is the membrane spanning domain. It is that this part is basically the transmembrane spanning domain. Then the cytoplasmic tail. Now, cytoplasmic tail is the domain which is actually causing endocytosis of the ligand receptor complex. So, these are the 5 domains of LDL receptor. Once again, one domain, first domain is important for binding of the ligand or lipoprotein. Second domain is for important is because it is actually causing expulsion of the ligand or lipoprotein inside the cell. Third domain is important for holding the receptor in such a pattern so that it can be bound with the coming ligand. Then membrane spanning domain, and finally the cytoplasmic tail or cytoplasmic domain, which is important for endocytosis of the receptor ligand complex. Now, LDL uptake apart from these tissues can be in macrophages as well. Now, LDL uptake in macrophages actually forming foam cells, which is causing atherosclerosis, which is important for heart attacks, myocardial infarction. We will discuss this in our next class in details. So, LDL receptor is important for uptake of LDL inside the cell. Now, how this uptake is basically occurring inside the cell? Now, remember LDL in low density lipoprotein, ApoB100 is the signaling protein which is sensed by the LDL receptor. So, whenever the circulating lipoprotein LDL comes in contact with the LDL receptor basically identifies ApoB100. Now, definitely there is receptor ligand binding, that is here ligand is the LDL, remember. So, this ligand is bound to the receptor, and that receptor ligand complex is endocytosed by receptor mediated endocytosis. Remember that LDL receptor is it is actually the surface receptor, and that surface is coated with clathrin. So, those clathrin coated pits are actually responsible for receptor mediated endocytosis. So, after endocytosis what is formed is endosome. So, you can see this is our endosome which contains, this is our LDL receptor, this is the lipoprotein LDL particle, and this part is our receptor. So, the ligand receptor complex is endocytosed within the endosome. Now, this endosome is fused with the lysosome forming endolysosome. Inside the lysosome there are different hydrolases. Hydrolases breaks down the cholesterol esters inside the LDL particles. Now, what happens? There is release of, you can see there is release of cholesterol as well as fatty acid, and also the cholesterol esters are accumulated, can be stored inside cholesterol ester droplets. Now, the apoprotein part is also hydrolyzed to amino acids, different amino acids, and released in cytosol, and there is amino acid, it is basically contributing to the amino acid pool of the cell, which is further reutilized for other protein synthesis. Now, remember in this receptor ligand complex, the ligand or the LDL is only hydrolyzed inside the lysosome, whereas the receptor, that is LDL receptor, is recycled back to the cell surface for further uptake of LDL particle inside the cell. Now, what is the fate of this cholesterol inside the cell? So, cholesterol, it has many biochemical properties, biosynthetic properties, like there are different steroid hormones which are synthesized from cholesterol, different vitamins like vitamin D can be synthesized from cholesterol, bile acids can be synthesized. Apart from that, it is one very important part of cell membrane, and also cholesterol is the cholesterol ester is cholesterol ester, they can be stored and can be utilized on requirement. So, this is the fate of cholesterol inside the cell. So, you can see that definitely LDL receptor is one very important signal for LDL uptake. Now, how this receptor is basically regulated? Remember, LDL receptor is regulated through cholesterol concentration inside the cell and as well as in the circulation. Now, what happens when there is cholesterol, when cholesterol is delivered inside the cell by LDL through the receptor ligand complex, that cholesterol can be stored or can be utilized for further different other biosynthetic requirement. Now, when there is when the cholesterol is delivered through circulation, the endogenous synthesis of cholesterol is stopped. So, this is a signal that the cell has adequate amount of cholesterol. So, the cell does not need to synthesize more cholesterol endogenously. So, the main enzyme of cholesterol synthesis like HMG coenzyme reductase is inhibited. So, endogenous cholesterol synthesis is inhibited. Now, there is a transcription factor which is known as SREBP, sterol responsive element binding protein. So, the sterol responsive element binding protein is influenced with the cellular intracellular concentration of cholesterol. So, when there is high level of intracellular cholesterol which is delivered through LDL, it basically is acting in such a way that the endogenous cholesterol synthesizing enzymes are reduced. Similarly, the LDL receptor synthesis is also reduced, which indicates that no more uptake of cholesterol is required. So, cholesterol receptor, remember cholesterol receptor synthesis is regulated with the, and that is signaled by the endogenous cholesterol intracellular cholesterol concentration through a pathway which is influenced by a transcription factor known as sterol responsive element binding protein. So, next we are moving to the metabolism of HDL or high density lipoprotein. So, if you remember the composition of HDL, that HDL is having the highest density amongst the lipoprotein, containing the highest amount of protein in the form of apoprotein. So, different types of apoproteins are present in HDL. So, basically HDL is acting as a reservoir for apoproteins in circulation. Now, HDL can be synthesized in liver as well as in intestine. Now, intestine contributes for around 30% of endogenous HDL synthesis. Now, the nascent HDL, once again just like VLDL and chylomicron, there is a nascent form of HDL and it has two different forms. One is hepatic HDL form, another is intestinal HDL form. Now, the hepatic form of HDL contains ApoA, apoprotein A, apoprotein C2 as well as apoprotein E. It is discoidal in shape. Why discoidal in shape? Remember, HDL does not have, this nascent HDL actually does not have triacyl glycerol which forms the lipoprotein core. Spherical core is formed by cholesterol, triacyl glycerol, but or hydrophobic lipids. So, those are not present in HDL. So, nascent HDL is basically discoidal in shape, and the surface of this HDL is formed by amphipathic lipids like phospholipids and free cholesterol. Now, the intestinal HDL contains only apoprotein A, no ApoC2 or ApoE. Basically, this ApoC2 and ApoE, these are synthesized in liver. So, the intestinal HDL, nascent HDL form does not have ApoC2 and ApoE, but it acquires when it comes to the circulation. When the nascent intestinal HDL comes to the circulation, it acquires ApoC2 and ApoE from the hepatic nascent HDL. So, this is the circulation or metabolism of HDL in our circulation. So, the nascent HDL or nascent HDL, say it intestinal HDL, say it hepatic HDL, they are mostly released in a form which is known as pre-beta HDL. Now, this pre-beta HDL mostly contains apoprotein A1. Now, what happens? Initially, it contains very meager amount, no triacylglycerol and very meager amount of phospholipid and cholesterol. Now, this pre-beta HDL, when comes to circulation, it is such a small particle, it circulates through the capillary, comes in contact with the extrahepatic tissues. Now, from extrahepatic tissues, there is transfer of phospholipid and cholesterol, mostly cholesterol from extrahepatic tissues to this pre-beta HDL. You can see pre-beta HDL uptakes cholesterol from extrahepatic tissues through the receptor ABCA1. So, this is a type of transporter protein ABCA1. ABC stands for ATP binding cassette transporter protein and the type is A1. So, this ABCA1, it is responsible for delivery of cholesterols from extrahepatic tissue to pre-beta HDL. Now, what happens when there is cholesterol and phospholipid accumulated in this pre-beta HDL? The enzyme LCAT is activated. Now, what that LCAT is? That is lecithin cholesterol acyl transferase enzyme. Lecithin cholesterol acyl transferase enzyme, as it is evident by the name that it transferase means it transfer acyl group from lecithin to cholesterol. So, you can see this is our LCAT enzyme. Now, in cholesterol, it is basically transferring the acyl group or fatty acid basically. So, here this part, the acyl group is transferred to cholesterol. So, there is esterification of cholesterol, formation of cholesterol ester, and lecithin is converted to lysolecithin. So, this is the function of lecithin cholesterol acyl transferase. Now, lecithin cholesterol acyl transferase or LCAT is actually associated with HDL and is activated by the apoprotein A1 mostly. Also C1, A4, they also stimulate the LCAT activity, but most important activator of LCAT is ApoA1, and remember pre-beta HDL contains ApoA1 in huge amount. Now, once this pre-beta HDL takes up cholesterol from extrahepatic tissues, what happens? This LCAT is activated by A1, ApoA1, and cholesterol is esterified to form cholesterol ester, which is more hydrophobic than the cholesterol. So, it is basically forming the hydrophobic core of in nascent HDL. So, basically nascent HDL is converted to mature spherical HDL form. Similarly, LCAT can also converts the cholesterol and phosphatidyl can convert the cholesterol of chylomicron and VLDL remnant to cholesterol ester as well. Now, once again another important protein is there, that is cholesterol ester transfer protein known as CETP. Now, CETP is another important protein with regard to HDL metabolism, and it is important for lipid transfer in between the lipoprotein molecules. How it transfer lipids? Basically CETP transfers triacylglycerol from either VLDL, LDL, IDL to HDL. So basically triacylglycerol is transferred from these to HDL in exchange of cholesterol ester which was in HDL is transferred to VLDL. So you can see from HDL there is transfer of cholesterol ester to VLDL as well as LDL, and what it takes? It takes triacylglycerol from VLDL or from LDL. So, this CETP or cholesterol ester transfer protein is important for transfer of triacylglycerol and cholesterol in between HDL and VLDL, LDL, IDL, these lipoproteins. Now, you can see CETP is important for reduction of cholesterol ester content of HDL. So, cholesterol was uptaken from extrahepatic tissue in the pre-beta HDL form. That cholesterol is converted to cholesterol ester with the help of the enzyme LCAT, and that formed cholesterol ester is transferred to VLDL or LDL with the help of the protein CETP. So, basically CETP is reducing the cholesterol ester content of HDL. So, again HDL is ready to uptake more cholesterol from the extrahepatic tissue. So, basically what you can see that CETP can influence the function of LCAT. So, more CETP acts, more cholesterol ester reduction from HDL, more LCAT is activated to convert the cholesterol to cholesterol ester. Now, CETP activity, when it is induced, CETP basically it transfer the cholesterol ester from HDL to VLDL or LDL, and they transfer it to the extrahepatic tissue. So, the requirement of cholesterol in extrahepatic tissue is inducing the CETP to transfer more cholesterol to the extrahepatic tissue. So, this is the function of CETP once again, that CETP transfers the triacylglycerol and cholesterol ester between HDL and other lipoproteins. Now, what happens for that? Remember pre-beta HDL, when it takes up cholesterol ester inside it, it is forming the hydrophobic core. Now, there is more maturation of the nascent HDL there, because there is more formation of cholesterol ester hydrophobic core, which is converting the nascent HDL, nascent discoidal HDL to the spherical matured HDL form, which is known as HDL3. Now, HDL3 contains cholesterol ester inside its core. So, this HDL3 is basically rich in cholesterol ester, but after CETP activity, CETP basically it reduces the cholesterol ester content of the HDL, whereas it increases the triacylglycerol content of the HDL. Now, after CETP activity, the form of HDL which contains more triacylglycerol and less cholesterol ester is known as HDL2. So, these are the isoforms of HDL, HDL2 and 3, which is, and this transformation is basically occurring through the CETP protein. So, once again we are back to the metabolism of HDL. So, you have seen that there is formation of pre-beta HDL. Pre-beta HDL, it takes up cholesterol from the extrahepatic tissue with the help of the transporter ABCA1, and it forms the discoidal nascent HDL form. Nascent HDL, it takes up more cholesterol ester from the extrahepatic tissue and cholesterol from the extrahepatic tissue, and that cholesterol is esterified with the help of the enzyme LCAT, forming the HDL3 isoform, HDL3 form of HDL molecule. Now, HDL3, remember, it is a spherical form. Now, HDL3 is converted to HDL with the help of the protein CETP, So, HDL2 is a triacyl glycerol rich form. Now, what is the fate of this triacyl glycerol rich HDL2? It enters the liver. Inside the liver, it is uptaken by the receptor SRB1, scavenger receptor B1. So, once it is uptaken, the triacyl glycerol is digested or degraded by the enzyme hepatic lipase. Remember, hepatic lipase, lipoprotein lipase needs ApoC2 for its activation, hepatic lipase does not. So, hepatic lipase here is degrading the triacyl glycerol content of the HDL. So, once the triacyl glycerol content of the HDL2 is reduced, basically it is forming HDL3, and once it continuously circulates through the liver, it is targeted by the hepatic lipase till there is total depletion of triacyl glycerol from the HDL, as well as also inside the lysosome there is degradation of cholesterol ester. So, finally, what is formed after action of after depletion of triacyl glycerol and cholesterol, there is reformation of the pre-beta HDL, remember, which does not have any triacyl glycerol or very minimal, very meager amount of cholesterol ester. So, this pre-beta HDL is once again formed for the cycle. So, you can see this is how cholesterol, the HDL functions. HDL is synthesized from liver containing ApoC2A and ApoE, and these apoproteins are donated for maturation of nascent chylomicron, for maturation of nascent VLDL. Then also it uptakes cholesterol from the peripheral extrahepatic tissues. Those cholesterols are converted to cholesterol ester with the help of the enzyme LCAT, and there is transfer of this cholesterol ester to VLDL as well as uptake of TG or triacyl glycerol with the help of the protein CETP. So, this is the function of HDL. It takes up peripheral cholesterol as well as it donates apoprotein for maturation of chylomicron and VLDL. So, this is how the dietary lipid in the form of cholesterol, chylomicron, and endogenous lipid in the form of VLDL are circulated, which is important for delivering cholesterol to peripheral tissue as well as triacyl glycerol to peripheral tissues, and this helped in their formation with the help of HDL molecule. So, in this session what we have learned is basically LDL and HDL metabolism. So, LDL is formed from VLDL remnant, and it is a cholesterol rich molecule containing ApoB100. Remember, this is very important. Apoprotein which is in LDL is ApoB100. The uptake of LDL is through LDL receptor, and then again HDL metabolism. This HDL molecule is a apoprotein rich particle which continuously takes cholesterol from the peripheral tissue, and it is important for exchange of triacylglycerol and cholesterol in between with other lipoprotein molecules, and also HDL acts as the reservoir of different apoproteins. So, these are my references. Thank you everyone. See you in the next class of lipoprotein metabolism.